Hi, Robert. It's Glenn. These are 2 boxes of prepared slides, and please handle with care because these buckles are come off by themselves very easily, and they're the only thing that's holding these boxes together. Sometimes for transportation, I use tape, but now that I'm here, I've removed the tape so these can fly open and the slides can come out and, fall very easily. I don't think the slides are actually in order, but this list represents what's here, but not necessarily in this order. Now, these are mostly slides of tissue samples from plants and animals, but that is not exactly what you asked for. So, I have 2 more boxes here, which is more apropos. These boxes have multiple repeats of the same slide. Let me show you what I mean. So in here, there is no labeling, but every slide has its own label. Here, let me get one here for you. So this guy, here I have paramecium. Yeah. Ooh, this one's conjugating. So there's a bunch of protists in here, and there's also some Volvox. So I've got, so if you see one of them that's paramecium, that means there's like 12 of them that's paramecium, all right? How about this guy? This is another paramecium. So this box might be mostly paramecium. Now I've got Stentor in the other box, and Volvox, as I said, and there's an amoeba in here. Yeah. There's some amoeba in here also, and there's also Vibrio cholera, so there's some pathogenic protists in there also. Vibrio cholera is a protist, right? I forgot. But here we've got, Vorticella, yeah. Is that pronounced Vorticella? I'm not sure. But here's that Volvox that I was telling you about. I got like a dozen Volvox in here. And there's some weird ones in here, like there's some tissue samples, some ferns. And I had something really interesting. Oh yeah, the Stentor is right here. So you just, if you're looking for the protists, I got dinoflagellates, the red tide guys. Yeah. There's a whole bunch of dudes in here. They're mostly Stentor in this particular box. Now these 2 boxes also, this is the only clasp that holds them together, so holds them closed. So please have your kids handle with care because it's very easy to drop all the slides at once as they just fall out if the box is being held upside down or something like that. I wonder if I can spot the amoeba for, oh yeah, this is the amoeba, nice. There's a couple of amoeba. I don't have like a dozen amoeba. I think I might have 6 amoeba. I'm not sure. But anyways, that's what you asked for. And here are the, I'm putting, so wait, I'll put these in I'll show you where I'm going to put these guys, these microscope slides. Oops, sorry. The microscope slides. Well, one of these slides is stuck. Okay, now that's better. All right, so I'm going to close this up, put that loose hasp on. Okay, now I'll put the microscope slides over here next to the biomaterials. I'm going to put it away from the edge of the counter so that I don't have a catastrophic fall there. I'll just stack them right there for you so you could easily see where they are. Okay. All right. Now, see you have the 3D Molecular Designs protein kit there. That's a cool thing there. Those are the same guys who made the water molecules. Those magnetic water molecules that show the intermolecular forces. Okay. Now, this is the animal cell. There are 24 parts in there, and they all come out. So you can take this right off the stand, and this is in 2 parts like that, so that comes off. It's really hard to put them back together, but it's all silicone or some kind of plastic, so you just wrestle with it a little bit and they come together. And every part in here comes out. There's a guide for what the manufacturer intended each part to represent right underneath the stand there. Okay. Now I've got the plant cell is similarly arranged. Now everything here is removable. So you can remove the cell walls of the neighboring cells like that, and then this this cell wall is also in two halves, so that can come off, and every organelle in there can also come out. This window, it's not necessary to remove the window, but this window is also removable. You don't have to remove the window because this cell wall is in two halves and it just comes out. Alrighty, I'll leave this here for you. Now, just as an extra, I brought a virus. So this looks kind of like a human influenza virus because it has the membrane envelope. Now, if you open it, if you open this, you'll find the capsid inside. Oh, it's easily opened there. And if you open the capsid, you'll find the a little blob of plastic, which is supposed to represent the genetic material. The virus is a little tricky because it has little pegs, little plastic pegs and little plastic holes in here. And this one, I don't know where the holes in, oh, here they are. So, so these plastic pegs need to go into the plastic holes if you want to put this back together. So I'll put that back together for you after this video. It's important to know, to note that the virus, of course, is not to scale compared to the eukaryotic cells. You wouldn't even be able to see the virus with a microscope, whereas you could see those easily with a microscope, in some cases, even without a microscope. Here's a nice bacterial cell with a nice flagellum here. If you open it, all we got inside is the chromatin, or the genetic material. Ribosomes would be really small. I don't know if they're modeling the ribosomes here. I didn't notice any myself, unless it's these green spots. But the virus is supposed to be a lot smaller, like 10 or 100 times smaller than the bacterium. And the bacterium is supposed to be 10 to 100 times smaller than the eukaryotic cells. But just brought these here in case you wanted to discuss those with your class. Warning: I don't know what the glycoproteins are prone to fall off, so please handle with care, even though it is plastic. Thanks very much. Bye.